Hello, my name is Juri Minxha, and I'm a postdoctoral scientist at the Cedars Sinai Medical Center in Los Angeles. The work that I'm going to be talking about today is the culmination of collaboration between Cedars Sinai, California Institute of Technology, and the Center for Theoretical Neuroscience at Columbia University. This work looks at representations of memory and choice in the human brain, and to the extent that these representations are modulated by the context or the task that the subject is performing at any given time. So I'd like to start with a few simple considerations. Imagine that you're looking at an image. This could be an image that you took while on a family vacation, or it can be a completely new image. In any case, we know that there must be some sort of internal representation that arises from the viewing of this image. And you could say that the goal of most of what neuroscientists do is to try to learn what this f of x actually is. What we're actually trying to do here is not only measure these representations that of visual representations and representations of familiarity that a person might have with this image, but also how these representations interact with the task that the person is currently engaged in. For example, you may be wondering if this is an image of a train, or if this is an image that you're familiar with. In either case, you're looking for very specific information that you're trying to extract from this image. In this particular study, we're going to focus on two specific kinds of tasks. In one case, we'll ask subjects a categorization task. We'll show them an image, and we can ask them, is this an image of a fruit, or is this an image of an apple? In a different cluster of tasks, we'll ask the subject if they've seen this particular image before. In addition, we'll show them images of a variety of different, that are sampled from a variety of different image categories. For example, fruits, faces, cars, and monkeys. And to reiterate, the goal of this entire study is to determine the extent to which these visual representations of these images are modulated by the task that the subject is currently engaged in. To address these questions, we designed a very simple task. Over a series of blocks, in this case, 8 blocks, we asked the subject to do one of these particular tasks. Every block is made up of 40 trials, and on each trial, the subject must remember the task that they're currently performing. They're instructed at the very beginning of the block what the task they're going to be performing in that particular block. In addition to the task instructions, they receive instructions about response type. So they can respond either by making a saccade to the left or to the right, or by pushing a button, yes or no. The neural data that we collect as subjects are performing this task is single unit spiking data. This data is collected from patients that have intractable epilepsy. In the top right, you'll see an anatomical scan from an example patient. The highlighted areas in green and in red demonstrate the location of example electrodes that are targeting the amygdala and hippocampus in this particular patient. In addition to the local field potentials that we record from these electrodes, we can also extract spiking activity. And this is the basic currency for most we're looking at today. In a single session, we can record from a variety of different areas. The areas that we're going to focus on here include the medial frontal cortex, and specifically the pre-SMA and the dorsal anterior cingulate cortex, as well as the hippocampus and the amygdala and the medial temporal lobe. In a typical session, we can record from up to 120 cells or 130 cells. On the right, I'm showing an example raster plot that evolves of the activity over a period of 10 seconds from all of these cells in a single session. And what you might be able to appreciate that in response to these different images, as we're showing them to the subject, there are these volleys of synchronized activity throughout the brain. On the right, I've highlighted the areas from which these cells are recorded. And in addition, note that some of these areas have a gray background and some don't. This indicates that they're recorded from the left or the right hemisphere. So in this particular task, we're recording from these two large structures in the brain, medial frontal cortex and the medial temporal lobe. We are very interested in the communication and the degree to which activity in these two areas is synchronized during the memory task and during the categorization task. Now, in addition to their scientific interest in this pathway, this communication between medial frontal cortex and the medial temporal lobe, there's a lot of clinical interest in this particular pathway, since it's being implicated in a variety of mental disorders, including schizophrenia and Alzheimer's disease. Our hypothesis in this particular study is that this pathway and the communication between these two areas is upregulated when the subject is performing a memory task. So, without further ado, we'll look at some of the population responses as the subjects are doing this task. And we'll start with the single unit activity that we observe in the medial temporal lobe, specifically in the hippocampus and in the amygdala. The population response as the subject is doing this task in the medial temporal lobe is actually quite varied. A lot of the cells that we record from are visually selective. What that means is that given the object categories that we use in this particular, that the images are sampled from, and that we use in this particular task, they are strongly selective for a specific object category. In this particular case, and this cell that you see here on the right is strongly tuned to images of monkeys. In addition to these visually selective cells, we also find a lot of memory selective cells. So these cells do not encode anything about the visual, but the category of the object that is on the screen, but rather whether the object that the subject is currently viewing is familiar or is novel. Note that this is a signal that can only arise throughout the duration of the task. This is not something that exists a priori. As subjects become increasingly more familiar with a subset of the images, the strength of tuning in these particular cells also increases. In addition to these two basic types of tuning, we also see a lot of cells that have mixed selectivity. For example, we might see cells that report the familiarity or the novelty of an item, but specifically for a object categories, for example, faces. So, given that there's so much heterogeneity across the population, perhaps there's a better way to summarize all of the responses that we see. Instead of showing you individual cells, what I'll do instead is accumulate the responses across all of the cells, as shown here in this heat map. And I'm going to plot the average condition. I'm going to calculate the average activity for each one of those cells for one of the 8 possible conditions. And these conditions correspond to the Cartesian product of 3 binary variables. First binary variable is whether an item is novel or familiar. The second binary variable is the task that the subject is actually performing when they see an image. In this case, whether they're performing an object recognition task or categorization task, or if they're performing a familiarity detection task. The third binary variable is what the object category is. So in general, we sample from 4 object categories, but for the purposes of this illustration and to drive the point across, I'm going to reduce it and only consider images of faces and fruits. Incidentally, it just so happens that their memory and their overall memory performance on this task is best for these 2 image categories. If we now look at the condition averages, the average activity across the entire population for these age conditions, and project this matrix into a 3-dimensional space, we actually recover a really nice geometrical representation. So each one of these condition averages here, marked as 1, 2, 3, and 4, corresponds to one of the conditions on the left. The planes that I've highlighted correspond to task 1 and task 2, blue corresponding to instances, the 4 conditions when the subject is in a categorization block, and red corresponding to instances when the subject is performing a memory task. What I've also plotted in this 3-dimensional space is a plane that divides items that are new, conditions that correspond to new instances when the images are new, from instances when images are familiar. I can also plot a hyperplane that separates instances when the item is a face and instances when an item is a fruit. There's a couple of things that I'd like to point out in this kind of arrangement, in this kind of geometrical structure. Let's focus on being able to decode whether something is new and old in only a subset of the conditions. So I've highlighted here condition 1 and condition 3. These correspond to a new face and an old face, or a familiar face. Note that a decoder that's fit to actually read out the familiarity information from these 2 conditions generalizes very nicely to instances that have never been seen before. So it can also report the familiarity of whether an image of a fruit is familiar or if it is not. This kind of generalization does not only work across images, but also works across tasks. Note that training a decoder in one task, for example, in this case, training a decoder familiarity information during the categorization task generalizes very well into the memory task. The same thing is true for visual information. We can train a decoder in one task. Here, this corresponds to conditions 1 and 2. So we can train a decoder to read out whether something is a face or a fruit in the categorization task, and we can test whether this generalizes to the memory task. And we can see that this kind of geometrical structure and this kind of representational scheme allows for very nice generalization across variables, and most importantly, across tasks. This, however, is not true everywhere in the brain. So what I've shown you so far is data from the medial temporal lobe, and I've reproduced that data in the graph that's in the top left here. We can see the same nice geometrical structure we were just looking at. If we were now to do the same exact analysis, but for data recorded in the medial frontal cortex, we see that while this information is highly decodable, it is much more distorted. We know this is decodable because we can actually perform this analysis, and that's what the bottom two graphs actually show. They show decoding in the medial temporal lobe and the medial frontal cortex with respect to image category in the left plot, and with respect to familiarity in the right plot. Notice that we've also plotted the decoding performance separately for each category, separate for each task. What we can already appreciate is that while the decoding values that we get within the medial temporal lobe for both image category and familiarity are mostly stable across tasks, in the medial frontal cortex, there is a great degree of variability as a function of task. The critical analysis here, though, is to test for generalization. And the way we do this is by fitting a decoder for either familiarity or for the actual visual category of the item that's on the screen in one task and testing how successful is this classifier at reading out information that is same information from the other task. So in this particular case, we're going to fit a decoder in one task and test in the other. And that's exactly what we do. And what we find is that while in the medial temporal lobe, the coding for both image category and familiarity is largely stable. And by stable, we mean that the same decoder fit in one task works very well in the other task. In the medial frontal cortex, it is the ability to decode this information is significantly reduced. A better way to summarize all of this information is with a metric that we call the generalization index. This is essentially a metric that measures how decodable the information is within task and normalize this with the ability to decode the same information across tasks. What you will appreciate from this plot is that while for both memory, in this case by mem, I'm referring to familiarity, for both familiarity and for image category, in the medial temporal lobe, we can decode this information very well, and the generalization index is close to 1. In the medial frontal cortex, the generalization index is significantly below 1. So these kinds of geometrical structures and these kinds of representational formats that allows for generalization across tasks and across many different kinds of variables arises in a lot of other different situations. Probably just what I find particularly interesting is that this kind of coding scheme also arises, for example, in word embeddings. In this now seminal paper from 2013 that looks at semantic embeddings of different words, we can find that concepts such as whether something is male or female are abstract with respect to the actual things that we are looking at. For example, the coding vector between king and queen is parallel to the coding vector between man and woman, suggesting that this concept of whether something is male and female generalizes well and is not specific to a particular instance. The same is true for example, for verb tense and country capital. So these kinds of formats are very similar. This kind of representation scheme is very similar to what we find in the medial temporal lobe for familiarity and for image category, where the coding vectors between things that are familiar and things that are novel are generalized well across tasks and across any of the task variables. So far we focused on representation of familiarity and representation of image category in the task. But what about choice? For that, we're going to zoom in on the population activity in the medial frontal cortex. So the medial frontal cortex shows in this particular task has a great degree of tuning for choice. I'm showing you here the raster plot for an example cell across all 320 trials, aligned with the onset of the image. Now, if we were to split this raster plot into instances when the subject was performing a memory task or a categorization task, we'll see that there's a very strong preference for memory trials. And if we were to split this raster plot even further into trials where the subject said yes or no, and note that in this case, yes and no correspond to something different. In categorization trials, yes could be an answer to a question of the type, is this an image of a fruit? And yes in the memory trials is the answer to the question, have you seen this image before? If we were to split now furthermore by choice, we'll observe that not only is there choice coding, not only is there task coding, but there's also choice coding. And that choice coding is specific to one particular task, in this case, to the memory task. There's a lot of heterogeneity in the population, and much like we see choice coding specific cells that code choice specifically for the memory task, we also see cells that code for choice in the categorization task, as is shown here. So what we'd like to know is how the representations of choice are affected not just by the task, but also by this instruction for response type. So note that there are four contextual signals. There are two contextual signals that the subject must always remember when they begin a trial. The first is what task they're currently in. Are they answering a memory question or a categorization question? And the second is the instruction regarding the response type. Are they communicating their choice with a button press, or are they communicating their choice with a saccade to the left and to the right? I'm going to summarize the data across the medial frontal cortex in a similar way as the familiarity and image category information in the medial temporal lobe. What that means is that I'm going to take all of the average activity across all of the cells in the medial frontal cortex, and I'm going to project it into a three-dimensional space so that we can get some intuition for exactly what changes, or get an idea of how this representation of choice changes as a function of response type and for task type. So these 8 conditions correspond to the Cartesian product of 3 binary variables. In this case, the binary variables are different than before. The binary variables in this case correspond, the first corresponds to choice, so yes or no. The second binary variable corresponds to task type, categorization or memory. And the third binary variable corresponds to whether the instructions were for, the subject was instructed to use a button press or to use a saccade. The 8 points that are plotted, the 8 condition averages that are plotted here, that are projected in this three-dimensional space, are exactly the same in the top graph and in the bottom graph. What's different is that in the top graph, I've highlighted the planes that connect all conditions when the subject was performing a button press. And in the bottom plot, I've highlighted all of the conditions, or I've labeled all of the conditions where the subject was performing a categorization task or a memory task. You can already begin to appreciate that there's a great degree of correspondence in the location of these condition averages as a function of a change in response type, and a lack of correspondence when there's a change in categorization. But we can when there's a change in task type. We can summarize this even better with a cross-condition or cross-task decoding performance. So if we train what these graphs show is training a decoder to read out choice information in one particular instance. In this case, in the top, you can see we're training on one particular type of response type. For example, training a choice decoder only on button press trials and testing its performance on saccade trials. In the bottom, we do something similar, but instead of training the conditions of relevance here are the task types. So we train on either categorization and test on memory, or train on memory and test on categorization. And what's very clear is that while there's very strong generalization the way that choice is represented across response types, there's a significant lack of generalization in the case when the task changes. So far we've looked at populations of cells within an area, and we've looked at representations of familiarity, we've looked at representations of visual category of the item, and representations of choice as a function of response type and as a function of task type. One thing that we're particularly interested in is looking at the communication between medial temporal lobe and medial frontal cortex as subjects perform or alternate between these two different task types, the memory task and the categorization task. And recall that our hypothesis was that this pathway, this synchrony, the synchrony level of synchronization between these areas is going to be significantly upregulated during the memory task. Now, there's many ways of measuring synchrony in simultaneously recorded in experimental settings where data from two different areas is recorded simultaneously. For example, you can look at synchrony between field-field recordings, or you can look at spec-spec correlations. And we performed all of this analysis. But in the interest of time, in this particular presentation, I'm going to focus on the results that we got from an approach that mixes across modalities. So in this particular case, we're going to look at a metric called spike field coherence. Specifically, we're looking at the degree to which spiking activity in the medial frontal cortex is coordinated by oscillations in this distal area, in the medial temporal lobe. So for every single cell, there's a number of different combinations that we have with this distant area. Recall that every single electrode that's inside in an area has a bundle of wires. So we can actually compute spike field coherence or synchrony between the activity of the cell, the spiking activity of the cell, and the oscillations in this distal area with many different electrodes. In this particular case, we have 7 possible combinations of cell and electrode possibilities. I'm showing here an example cell in the medial frontal cortex, and specifically in the dorsal anterior cingulate cortex, and the degree to which the activity of the cell synchronizes with oscillations in the hippocampus across these 7 electrodes. At the very bottom of this plot, you'll see that I've split now this metric of pairwise phase consistency as a function of the task that the subject was performing. What you can already appreciate is that this synchrony at roughly 7 hertz or so, which is in the theta range, is much stronger during the memory task than it is during the categorization task. And across all possible cell electrode pairs, in 33 sessions, we had 8,822 cell electrode pairs with which we can actually do this analysis. We see that there's very strong modulation of synchrony, specifically in the theta band, with a peak at 5.5 hertz. What is not shown here, but is actually available in the published version in manuscript, is that not only is this effect theta specific, but it's also cell type specific, meaning cells that are much more strongly tuned to that represent choice specifically during the memory task have a much stronger coherence with oscillations in the medial temporal lobe than those cells that encode choice specifically during the categorization task. So this hypothesis, if you recall, we hypothesized that the inter-area coherence between MFC and MTL is much stronger during memory retrieval. What I haven't shown you here is that the degree of the synchrony is also predictive of correct memory retrieval. And if you're interested in seeing those results, I'd like to refer you to the published manuscript. So I'd like to leave you with a video summary of all of the things, of some of the key results that we've seen so far, and I'll describe to you exactly here. So we've designed this task that asks the subjects to alternate between either a categorization question or a memory question. In both instances, the subject reports produces a binary choice saying either yes or no. What we observe is that the choice activity or the choice representations occupy different subspaces during the categorization task and during the memory task. In addition, we also find that during the memory task, In addition, we also find that during the memory task, that the activity of the spiking activity in the medial frontal cortex is very strongly regulated by theta oscillations in the medial temporal lobe. So on that note, I'd like to thank you for your attention, and I'd also like to thank all of the people that work. Specifically, I'd like to thank the Rutishauser Lab and my advisor, Ueli Rutishauser, the ADOS Lab at California Institute of Technology, and Stefan Fuzzi at the Center for Theoretical Neuroscience at Columbia University.